Today's lesson is on passive and active transport. And this lesson, I just want to apologize. I forgot to put the notes in the book that I made for the grade 10 teachers, and therefore I'm making this video. Your teachers did explain this to you, but I just want to do a little bit more detail. I do apologize to the teachers. I'm sorry. So this is a nice diagram to show you, to demonstrate passive and active transport. So when you're in the bottom of the hill, that's a low concentration. And I always use the brackets because that's a sign symbol for concentration. And at the top, that will be high concentration. So my arrow here means high and that will be low. Now, if you imagine going up a hill, if you're going up a hill, so let's do it this side because that's how the diagram is doing it. If you're going up a hill, it's very hard to actually run up a hill. You need a lot of energy and you need a carrier. And if you remember from your cell membrane, the carrier is the protein, okay, which your teacher did explain to you. When you're going downhill, so active transport is when a substance moves from a low concentration to a high concentration. You need energy and you need a carrier. The energy, you guys should know, comes from the mitochondria. Passive transport is when a substance moves from a high concentration to a low concentration. You don't need energy and no carrier. Because if you're going down a hill, all you have to do is sit down and just slide down. So it's a bit easier. So that I just want to explain the diagram, the picture, if you'd like to use that. Now, very important terminology you have to know is a concentration gradient. And that's in the difference between concentration. That means if you have a high concentration and a low concentration, that's a gradient. It's a difference in the concentration. If you have a high and a high concentration, that is not a gradient, that's not a difference in concentrations. Right, so molecules always seem to move up down a concentration gradient. It's always easy to move from a high to a low, right? So you always move from an area from high concentration to an area of low concentration. And the example we use is the food coloring. So imagine if this is food coloring here, you put a drop of food coloring and eventually, so this is the area of high concentration of the food coloring. And this is the area of low concentration of the food coloring, and you can see what happens. The food coloring moves from a high concentration to a low concentration until evenly spread. Okay, well, that is just an example of diffusion, which you also learned in grade 8. Okay, selectively permeable membranes, you already learned this. Selectively permeable, semi-permeable, differentially permeable, partially permeable is when a membrane, example, the cell membrane is selectively permeable, allow certain substances to enter and certain substances to leave. And here the example is water and oxygen. And now osmosis. Osmo means water. When water moves from a high concentration to a low concentration through a semi-permeable membrane. So this is your explanations for important terms you need to know. Now let's look at osmosis. Now, here we talk about a hypotonic solution. A hypotonic solution is a solution where there is less salts. In your case, you learned solutes, also known as more diluted. So you can use whatever word makes you feel happy. Hypertonic is more concentrated. And there's more solutes or salts. Okay, so I'd like to explain it with my own explanations that I use. So if I'm drawing, let's say, in a hypotonic solution, so here you have your beaker with your water. It's a hypotonic solution, very, very, let's just say it has 1% of salt. And then you have your potato. The first thing you must do is you must write, so there's your potato, and you can even try to do this at home. So the first thing you must do is figure out where there's a high concentration of water. And high concentration of water means high amount of water. So high concentration of water is here in the beaker here. Potato, of course, you learned the vacuole has sugars and salts. So there's a low concentration of water. Do you have a gradient? Yes, you have a difference in concentration. The potato is a living cell, so all living cells have a cell membrane. So water moves from the from the water, the beaker, into the potato. And when water moves into any cell, what's going to happen to the cell? The cell is going to swell. Now that is osmosis. But because water is moving in the cell, we call that endoosmosis. And this is the same principle when the plant is absorbing water, when the roots are absorbing water. When water enters the root hair cell, it is endoosmosis. So if you write osmosis, you get 1 mark. If you write endoosmosis, you get 2 marks. So you can take a picture. I'll give you a few minutes to take a picture of this if you want to, or you can replay it. You can also try this at home. When you put potato in water after you peel it, you will notice after a while the potato swells. So let me move on to the next one. Cleaning this. The next one we're looking at, and I like to talk about potatoes because we've done many experiments with the potatoes, and so it's easier to use. Sometimes it's easier to do simple experiments at home so that you can understand the osmosis. And so the next one we're doing is a hypertonic solution. Hypertonic we know is more concentrated. Now, here I'm going to draw my beaker again. My water, there's my potato. You can also go home and try it at home. Only thing is now, here it's going to be a salt solution. And let's make it a 20% salt solution. So that means in this salt solution, there's more salt than water. So immediately that's a low water concentration. In the potato, you know there's a vacuole with water and there's a higher concentration of water. Is there a gradient? Yes, there is a gradient. We already know the potato is a living, so it has a cell membrane, and which is semi-permeable. So water moves out from the potato into the into the sugar salt solution. Water moves out of the cell, so the cells will shrink, and that is called, and that's called exosmosis. And again, if you say exosmosis, you get 2 marks. If you say osmosis, you get 1 mark. So that is now the explanation of osmosis that I usually use for my class, and I prefer to use example with a potato because sometimes you get experiments like this in the class or in the exam. I'm going to give you a few minutes to take a picture if you want to. And then we can go back to our notes. So there we are here. It talks about water moving into the cell. That's endoosmosis. Water moving out of the cell. That's exosmosis. In plants, the same thing happens. If you take a normal plant, that's a normal plant. There's the nucleus, there's the cytoplasm, and there's the vacuole. So in a normal plant, this vacuole is very firm with water, and you can see the cell is firm because the vacuole maintains the shape. So as the plant starts to dehydrate, you can see the vacuole loses shape. The cell membrane moves away from the cell wall, and you can see the cytoplasm and the vacuole shrinking. And this is called plasmolysis. This is also known as when the cell becomes flaccid as well. And this is usually what happens when you don't water your plant, or if you put a plant in, if you water the plant with sugar water, salt water, which we won't do, it mostly happens with dehydration. To cause this cytoplasm and vacuole to become firm again, all you do is you put the plant or the cell, you have normal tap water that you can actually use to make the cells firm again. The same thing that happens with lettuce. When lettuce becomes limp, the vacuole loses water. And again, if you put the lettuce in cold water, just cold plain water, tap water, you'll see it becomes firm and crisp again. So that is now Passive transport, that was osmosis. And we've done diffusion with the food current. With diffusion, it's the movement of any substance besides water from a high concentration to a low concentration. With diffusion, you don't need a semi-permeable membrane. Active transport, you will learn this in grade 11. It's against the concentration gradient, meaning when a substance moves from a low concentration to a, excuse me, girls, from a low concentration to a high concentration. That's also known as against the concentration gradient. It's like swimming against the tide. And very important, it's the proteins that will carry and allow the substance to move in. This is an example here. There's the outside of the cell, there's the inside of the cell. So here, the substance is going from a high concentration to a low concentration. You can see it goes against the gradient. For this process, you need energy, and the energy comes from the mitochondria. And there you can see the substance actually entering the carrier, the protein. And the substance enters the protein and then goes from the outside of from the inside of the cell to the outside of the cell. It's almost like a passage. You will learn more about this in grade 11 when you learn active transport. Thank you.